name is Tom Jessell, and I work at Columbia University in New York City. So for a long time, the research that we've been focusing on has attempted to understand the neural circuitry that controls movement, since all aspects of expressed behavior depend on the ability to move muscles, from facial expressions to running in terror from predators. Movement is an essential feature of all animals, and people in fact have argued that the only thing that distinguishes animals from plants is the ability of animals to move. And so we're trying to understand the circuits in the spinal cord and brain that give animals the ability to control their muscles with fidelity in a way that produces behavioral responses. So the lecture that I presented this morning was an illustration of how complicated the brain and nervous system really is, because you can use contemporary molecular genetic approaches to study one neuron as part of a motor circuit, in the full knowledge that there are 100 or 200 other classes of neurons, and an eventual understanding of motor circuitry is going to depend on an appreciation of the coordinated action of 200 of those sets. And yet when you dig in for one class of neuron, you realize how complicated it is to arrive at any information that you have confidence in. So the nervous system is complicated. Presumably it needs to be complicated to adapt to the variety of insults that the world slings at it, and still to survive. And modern techniques in neurobiology and physiology and molecular genetics are beginning to reveal that complexity in full magnitude. So the talk today was really just studying one set of inhibitory neurons that control sensory motor transmission, and trying to build on the classical work of physiologists some 60 years ago to derive behavioral correlates of the manipulation or activity of that one set of neurons. 100 years ago, because methodologies didn't exist with the sophistication of today, people were working with whole animal, organismal and tissue problems. I would say that the last 50 years has seen a veering away from that towards understanding the properties of single proteins and single molecules that are of relevance to neural function, and great progress has been made in that. But now we're beginning to try and reconstruct, with that molecular expertise and experience, the classical problems that were first formulated 50, 100 years ago. So the field has moved full circle, but arguably with a better sense and better set of reagents and methodologies to approach problems that people appreciated 100 years ago but didn't know how to tackle. Why come to an international congress of this sort? I mean, there are many reasons. One is the marvels of Birmingham. The other is that because science is complicated and the study of any given system is complicated, one is inadequate individually to cover all of the aspects of a complicated problem like movement. So coming to a conference like this gives one the opportunity to talk to people who are working on the same problem from very different perspective and very different set of backgrounds, and try and learn from them and educate oneself in ways to approach this problem. I mean, because of our interests, I seek out the physiology of motor systems, and particular people who are studying aspects that we don't think about so much. So there is sort of integrated aspects of motor control, particularly the way the brain controls the spinal cord. We've been digging around in the spinal cord for more years than I care to remember, and the spinal cord is, in a sense, the servant of plans and commands that are coming from the brain. So understanding that link, I think, is one of the futures, and digging around posters and talks to try and sort of help frame a set of questions for that issue. Based on all the mistakes I've made, how would one provide advice for a young person interested in this field? I think the most important thing is to be passionately enthusiastic about one topic and tenacious enough never to let go of it despite the adversities and disappointments hurled at you on a daily basis. But if you're interested enough, then the knowledge that emerges and accrues from actually making a little discovery that people didn't understand before, it's kind of cool. And so you have to resonate with that view, that you're doing something that is brand new, you don't know how to do it, you'll make mistakes, but eventually something will work. It'll never work as ambitiously as you thought of going into it, but you have to take solace from small victories. Well, thank you, Quentin, for that generous introduction, and it's a real privilege to be here to have the opportunity to talk to this diverse and distinguished society. It's a particular pleasure to have the opportunity to present the T.P. Feng lecture. Not sure how many of you know of T.P. Feng and his accomplishments, but I want to present him in the context of IUPS, where he served on the council. And so here is T.P. Feng a few years ago, surrounded by many luminaries. Here's Andrew Huxley, some of my favorites. Jan Janssen is here, Rami Rahamimov, and Chris Krenovich. And so in this context of the relationship between neural and muscle, T.P. Feng was a pioneer for over 50 years. Early on, working with A.V. Hill, he published this paper on the metabolic and contractile properties of muscle absent neural stimulation, a phenomenon that later became known as the Feng effect. And throughout this career, he's maintained this interest in the relationship between muscle metabolism and contraction and its neural innervation. And so this is a field that I have been involved in, albeit somewhat peripherally, but nevertheless it's a privilege. And if you think about the people that Feng worked with, from A.V. Hill to Adrian to Sherrington to Eccles to Granit and to Lorenzo Deneau, this is a group of distinguished associates, and Feng clearly belongs right up there with them. And I also like to thank the Gatsby Charitable Foundation for generously sponsoring this lecture. So what I wanted to talk to you about this afternoon, is our attempts to examine the systems and circuits that give animals the ability to act on demand. That is, to process diverse information from the sensory world that surrounds them and divert that and transform that to the goal of purposeful behavior through movement. And if one stops to think about it, even the simplest of movements involves both a planning and executive phases and engages circuits at many levels of the neural axis. The planning phase, prior to overt action, involves diverse circuits that exist at supraspinal sites. But really, from the time of Sherrington, we've appreciated that the executive component of movement, that is deciding exactly which muscles are activated over which period of time and in which combinations, those challenges have largely been assigned to circuits that exist within the spinal cord. So in a sense then, the expression of behavior, if not the inception of behavior, depends on understanding the way in which the motor system activates muscles in precise manner, because behavior is nothing without movement. And if one moves into the spinal cord to try and examine some of the behaviors, then amongst the most sophisticated that exist in mammals are those involved in the control of limb movement. And here are two examples taken from the work of the Italian futurist Giacomo Balla, one indicating locomotion, the iterative movement of the limbs to move you from one location to another to avoid predators or for other purposes there, to the more sophisticated initiation of hand and arm movements used in reach and grasp and object manipulation. And in this talk, I want to focus on the neural control of this second aspect of reaching movements, because these are thought in higher mammals to represent some of the most sort of acquired aspects of human motor control here. And they involve extrapersonal sensory cues, they involve the proprioceptive control of limb movement, the knowledge of where your limb is in the environment, and they also involve some appreciation of the behavioral context of the intended motor act. And so limb movements involved in reaching and grasping are really
they are not guaranteed events in terms of accuracy and fidelity of output. Hence this quote from Byron to indicate the slight chance of reaching here. So this aspect of forelimb motor control has primarily been the product of studies in humans and non-human primates to understand the logic of circuits that control forelimb movement in reaching. And just to summarize a very complicated aspect of this field, the idea here would be that there is somewhere in supraspinal circuits a motor plan that is conceived, which then sends a series of descending commands which eventually will activate muscles. The muscles will then produce a sensory consequence through sensory feedback from the proprioceptive system in particular, which is fed back to relays and provides an update in this plan. Because during these reaching movements, there's thought to be constant refinement of limb trajectories to accommodate minor deviations or the change in the intended target here. So the problem with this circuit is that by the time this information is fed out to the periphery back through the sensory feedback system, it's arriving at these motor planning centers too late to effectively update an intended motor plan or motor command system. So the nervous system, it is thought, has evolved ways of overcoming this delay problem in feedback and refinement in a couple of different ways. So one way is that every motor command that is intended for motor neurons and muscle activation is thought to provide a copy, an internal copy, which then bypasses this peripheral delay system. So this would be an efference copy or corollary discharge system that is fed back to provide rapid updating of these motor plans. And a second way would be is that the sensory feedback information is subject to segmental spinal gating or control systems that again obviate the need for this long delay. But in both of these cases, either in locomotion or in reaching, the precise details of the circuitry involved in these motor control systems, the copy and gate systems, remains rather obscure. So these are the views derived from the primate studies of the motor system and raise the question of whether you can study these sorts of problems in lower mammals, in particular in rodents, in rats and mice. And since mice obviously offer the advantage of genetic manipulation, the first question is whether in fact rodents can reach in a way that approximates their mammalian counterparts. And Ian Wishaw in Canada has been arguing for some time that there are really very great similarities in the elemental aspects of reaching in humans and in rodents like the rat, that at many different stages, the lift, the advance, the pronation, and the grasp phase, the similarities viewed suggest that many aspects of this motor behavior have been conserved in evolution. And we've been interested in this, in trying to see whether this extends into mice and whether in fact you can train mice to reach for targets in an effective way and study forelimb motor behavior as distinct from the locomotor systems that have been primarily the pursuit of rodent interest in motor control. And so Ayman Azim in the lab developed a system based in part on Ian Wishaw's work in which a mouse is placed in a container. There's a small slit that separates the animal from a desired food pellet. And the job of this mouse is then to reach through this slit, grab the pellet, while at the same time performing high-speed video kinematic analysis. And then with a little bit of training, what you can find is that these animals do quite a good job of reaching for this target. And so this is now a video, and we've placed small reflective markers on the paw and on the target, and in this way you can actually demonstrate that mice, as in rodents, as in monkeys, are quite good at reaching for targets when encouraged to do so. So this provides a basic assay which we've been using now to interrogate neural circuits involved in reaching movements in mice. And the other thing that is important in this approach that we've taken is to argue that we would be better off in dissecting these circuits had we some more sophisticated way of manipulating individual classes of neurons in genetically defined ways and looking at the consequences for motor behavior. And so the developmental work that we've done has gone into great detail, as Quinton mentioned, in defining cell types in the spinal cord on the basis of their transcriptional provenance, and then using these transcriptional markers to build up aspects of the circuitry here. So what we're now going to do is try and use some of this information about the identity of motor neurons and interneuron types in the spinal cord to try and understand not only the organization and assembly of the circuits, but the way that these circuits function, looking at the way that different classes of excitatory and inhibitory interneurons might control motor output in the context of these reaching motor behavioral tasks. And so I'm going to give you just an initial glimpse of studies that we've been performing to look at the function of excitatory interneurons in the context of these internal copy systems, and then spend most of the talk talking about the role of inhibitory interneurons in spinal cord and one particular type of inhibitory interneurons in the control of reach. So if we begin with the excitatory interneurons, then this set of neurons, which turn out to be the V2A neurons shown in this circuit diagram here, are relevant, I think, in terms of the idea of a spinal internal copy system that is, in a sense, reporting to sensory processing centers in supraspinal sites about the activity of motor output systems. And so I'm really going to go through this rather briefly. This is the work of Ayman Azim, who developed this reaching task, just to give you a sense of the flavor of the sorts of experiments that one can do. And so prior work from Broer-Alstermark in particular had pointed out at cervical levels of the spinal cord in cat, but also now in rodents, that there's an interesting group of neurons called Propria spinal neurons that provide an excitatory premotor input to the motor neurons that are controlling forelimb muscles here, but through an axonal bifurcation also send axons ascending to the lateral reticular nucleus, a sensory processing center that feeds into the cerebellum. And so in a sense, in this one neuron, you have welded a premotor output and a copy internally that is projecting to the cerebellum, indirectly rather, lateral reticular nucleus. So this forms a spinal copy system here. And in work that I don't have time to go into in great detail, Ayman showed that these propria spinal neurons represent a major subset of the V2A neurons, which are defined by this CHEX10 transcription factor. So here are the neurons in the spinal cord that correspond to these propria spinal neurons. And with this genetic entry point, we can do a number of things. We can selectively inactivate these propria spinal neurons and look at the consequences for reaching behavior, or in particular, we can selectively activate this ascending sensory reporting branch of these neurons and see whether decalibration of the motor output from the sensory input branch produces any abnormalities in behavior. And I just want to give you one example that that is the case. So here we've expressed channel rhodopsin in propria spinal neurons and used a fiber optic laser light probe shining light at 473 nanometers to excite the terminals of these propria spinal neurons as they project up to this cerebellar processing center. And if you perform those experiments in relation to the normal faithful reach that I showed you earlier, then you see that there are profound abnormalities in reach. The animal is incapable of making a direct trajectory, is in fact rather incapable of getting its paw even through this perspex slit to reach its target. So these experiments, and others that I don't have time to show you, really begin to pick apart some of the excitatory interneurons that might be responsible for some of the internal copy systems that are reporting selective aspects of motor output. But I want to spend most of the time in the context of these reach behaviors dealing with inhibitory interneurons that are plausibly involved in this local segmental gating system here. So to introduce this, it's necessary to consider the range of inhibitory circuits that exist within the mammalian spinal cord, defined from classical physiology by Eccles and his colleagues over many, many years. And what we've learned is that there are many different classes of inhibitory microcircuits, and they can be divided into two major sets. The first, as shown here, are inhibitory neurons. These green things are inhibitory neurons that we're calling or grouping as postsynaptic. That is, inhibition is exerted directly through inhibition of output motor neurons. And there are many different varieties of these microcircuits. They're feed-forward disynaptic inhibitory circuits. They're reciprocal inhibitory circuits triggered by sensory input. These would be the group 1A reciprocal inhibitory interneurons from Lundberg and Eccles. And this would be a feedback system triggered by motor neuron. This would be the Renshaw cell, as defined initially by Birdsey, Renshaw, and Eccles and others. So this is one major class of inhibitory interneurons that exist in the spinal cord. But there's a second class of neurons that differ conceptually. And these are neurons that inhibit sensory motor transmission not by inhibiting the motor neuron directly, by exerting a presynaptic inhibitory influence on the terminals of proprioceptive sensory neurons that are forming contact with the motor neurons. So these are postsynaptic inhibitory neurons. This is a presynaptic inhibitory class. And studies over many years have revealed certain distinctions in principle in the function of post- and presynaptic inhibition. Postsynaptic inhibition is pervasive. That is, inhibition of the motor neuron will block motor neuron response to all of the many inputs to that single motor neuron, whereas presynaptic inhibition provides a selective gain control over one of the many inputs. Then postsynaptic inhibition is decreasing the probability of motor neuron spiking, whereas presynaptic inhibition is decreasing the probability of transmitter release. And then viewed in terms of information transfer, postsynaptic inhibition is termed to be subtractive, whereas presynaptic inhibition is divisive. So there are these fundamental distinctions in pre- and postsynaptic inhibition. So as I'm sure you know, the origins of presynaptic inhibition came about 55 years ago when Mike Fortis and Kay Frank and later Eccles defined the existence of presynaptic inhibition in spinal cord. So the type of experiment they performed is shown here. They record in cat from an extensor motor neuron, stimulate extensor sensory afferents, and record a large EPSP. If then they provide a conditioning stimulus of flexor afferents, which recruits via this intervening interneuron, these presynaptic inhibitory interneurons shown in red, then the amplitude of the EPSP is greatly diminished. And subsequent work showed that this was not in fact remote motor neuron dendritic inhibition, but it really reflected presynaptic inhibition of sensory input. One of the striking things about presynaptic inhibition in the spinal cord, as opposed to its counterpart in many regions of the brain, is that there is an anatomical substrate for presynaptic inhibition through the axoaxonic synapse. And so here at the electron microscope level, you're looking at a motor neuron, you're looking at a neurobiotin-filled sensory bouton, and here shaded in this sort of reddish hue is a P bouton defined by Conradi as the terminal of the GABAergic interneurons that exert presynaptic inhibition. And you can't really see it here, but this is a GABAergic bouton because you can see gold particles in this immuno-EM here for GABA here. And so this is an anatomical substrate for presynaptic inhibition that differs from the sort of spillover inhibition that you see in many supraspinal regions here. So then the question that we've been interested in, which is unresolved after these 60 years or so, is what purpose does presynaptic gain control have in reaching and motor behaviors? You could imagine that with all of these postsynaptic inhibitory neuronal systems existing in the spinal cord, presynaptic inhibition is just an evolutionary flourish that doesn't contribute much to the control of motor output. And part of the reason that this problem hasn't been resolved is that presynaptic inhibition uses GABA. Postsynaptic inhibition in part uses GABA, although as well as glycine. So conventional antagonists haven't been able to tease apart pre- and postsynaptic inhibition. And the second thing is that the diversity of interneuron subtypes defined physiologically don't have molecular labels that can permit interventional analysis. So in our studies, we've been interested in 3 general aims to probe the function of presynaptic inhibition. The first is to see whether we in fact can obtain genetic access to GABA pre-neurons as distinct from all of the postsynaptic inhibitory GABAergic neurons. And if we can do that, can we test rigorously whether these GABA pre-neurons, as I'll call them, whether activation of those mediates the classically defined presynaptic inhibition defined by Eccles and colleagues in CAT. And if these 2 things are true, can we then manipulate GABA pre-neurons and look at their contribution to motor behavior using this reaching task? So over the years, the last few years, we have in fact been able to provide a molecular signature for the GABA presynaptic neurons that distinguishes them from their postsynaptic counterparts. And this is the work of 2 people who were formerly in the lab, Nick Betley, a graduate student, and Julia Kalschmidt. And so this falls into the large group of studies we've been done, which are defining each of the maybe 100 or so neuronal cell types in the spinal cord on the basis of their transcriptional provenance. And Nick and Julia showed that the GABA presynaptic neurons derive from one progenitor domain defined by expression of a transcription factor PTF1A. And by using lineage tracing and following the settling position of those neurons, we know that all of the GABA presynaptic neurons that are seeking out the sensory terminals here are clustered in a small region just dorsal and lateral to the central canal. And we've been able to trace a sort of a preliminary pathway where this transcriptional provenance leads to a difference in the GABA synthetic capacity of these GABA pre-neurons as opposed to their post-neurons. And this relates to the expression of a GABA synthesizing enzyme, glutamic acid decarboxylase variant 2, a gene that is different from the GAD1 gene that is the major GABA synthesizing gene. So if we look at this in detail, as shown in this slide, we're looking at a motor neuron here, shown in brown, with a sensory proprioceptive input with a GABA terminal forming a presynaptic contact on that sensory terminal on the motor neuron, and here is a different GABA post neuron. And if we start using neurochemical markers of synaptic boutons, here is the motor neuron labeled with some fluorescent protein in a transgenic mouse. Here is a single elongated sensory terminal shown in blue, visualized by VGLUT1 expression. And you can see that this is studied with 5 or 6 presynaptic inhibitory boutons defined by the expression of GAD2. And if we do double labeling with GAD2 and GAD1, you can see that both GABA synthetic enzymes are expressed by these boutons on sensory terminals, which is quite different from the GABA post boutons. So here's a motor neuron, here's GAD1 in a GABA post neuron. There isn't a sensory terminal in sight, so we're looking at this region. And when you do double label, these have GAD1 but not GAD2. So GAD2 provides a unique distinguishing marker of the GABA presynaptic inhibitory neurons. And we reasoned that this might provide us with a genetic entry point to mark and eventually manipulate the GABA pre-neurons, leaving the GABA post-neurons intact. So Andrew Fink, a graduate student in the lab, has been pursuing this idea using genetics to try and mark GABA pre-neurons, look at their physiology and behavioral consequences. And so we were fortunate that at around the time we were performing this study, Josh Huang at Cold Spring Harbor made a GAD2 Cre recombinase transgenic mouse, which rather nicely recapitulates the pattern of GAD2 expression. So in this mouse here, in an adult, if we inject a recombinant adeno-associated virus encoding channel rhodopsin and a YFP reporter, then in those neurons that express Cre recombinase, there's a flip of this inverted reporter to produce effective expression of channel rhodopsin and YFP. And so if we inject this AAV reporter construct into cervical levels of the spinal cord here, what you find is fluorescent protein expression is nicely tagged to these blue VGLUT1 sensory terminals in the ventral spinal cord. This is the adult situation. But we wanted to perform the physiological studies in vitro at neonatal stages, and at this stage we run into a complexity with the pattern of expression of GAD2, in that at earlier developmental stages, GAD2, and therefore this GAD2 reporter mice, are expressed in many more GABAergic neurons, including some of the post. So over this period that we were performing physiology, we haven't segregated or sequestered into this selective marker for GAD expression in GABA pre-neurons. So we're going to have to use the physiology here to tease apart expression in GABA pre and GABA post neurons. So to try and do that, Andrew then developed this isolated hemisected spinal cord preparation to try and identify and manipulate the GABA pre-neurons. So this is just the proof of the ectopic expression, if you like. Here we're getting fluorescent protein labeling in the GAD2 terminals on a sensory neuron, but also this ectopic expression in these GABA post neurons, and this is the quantitation here. So in this isolated spinal cord preparation, we have marked GABA pre and a few GABA post neurons expressing YFP and channel rhodopsin here. We wanted to record from the GABA pre-neurons and then examine whether optoactivation by shining a laser light would activate channel rhodopsin and entrain the firing properties of these GABA pre-neurons. And we helped ourselves a little bit by focusing on this region where we know the GABA pre-neurons are located. And so if we do that, each of these blue bars here represents a brief light pulse at 473 nanometers, and you can see that you can very effectively elicit spiking in these GABA pre-neurons here, and these sort of raster plot displays show the fidelity of spiking with each light pulse activation. And this follows in a nice linear range at frequencies of light pulse activation between 1 and 50 hertz, and then plateaus at that point. So using this preparation, the ability to activate GABA pre and a few GABA post neurons, we wanted to run through these neurons in terms of the hallmark features of presynaptic inhibition defined in the cat. And so those features are, first of all, this association between presynaptic inhibition and the phenomenon of primary afferent depolarization, the depression of sensory evoked EPSCs and EPSPs, and the mediation of this repression through a reduction in transmitter release probability. So does optoactivation recruit and mimic all of these features observed in the cat? So primary afferent depolarization is this phenomenon which, in a sense, reflects the activation of inhibitory interneurons that release GABA on sensory terminals. And because of the chloride equilibrium potential in sensory neurons, GABA is depolarizing in those neurons. That depolarizing wave is transmitted backwards along the dorsal root, can be measured as a dorsal root depolarization or primary afferent depolarization. So when you stimulate in mouse one dorsal root, record from an adjacent dorsal root, you can see this depolarizing potential which reflects primary afferent depolarization. When you light activate the GABA pre-neurons under these same conditions, you elicit a depolarizing potential that is very similar in time course and amplitude to that induced by dorsal root stimulation. Moreover, the pharmacology indicates that this depolarization can be blocked by GABA antagonist and not blocked by strychnine glycine antagonist. So this has all the characteristic hallmark features of classically defined primary afferent depolarization, a surrogate for presynaptic inhibition. Does then activation of these GABA pre-neurons suppress EPSC amplitude recorded from motor neurons in response to proprioceptive input? So to approach this question, Andrew then modified this splice preparation where we could patch record from identified limb-innervating motor neurons, which are identified by GFP expression in a transgenic mouse, identified by filling with dye after recording. And if we stimulate the dorsal root, you can record an EPSC of very constant onset latency here. So the coefficient of variation of the onset latency here is remarkably constant and tight, an indication of the monosynaptic nature of this postsynaptic response. So this isolates the proprioceptive input to motor neurons and then permits us to ask what happens when you activate the GABA pre-neurons in this context. And when you do that, activation of these neurons produces a significant suppression in the amplitude of the EPSC, about 40 to 50%, depending on the experiment. So this is very reminiscent of the suppression of EPSP amplitude defined by Frank and Fortes and by Eccles. Now, in principle, because we're activating the postsynaptic neuron as well as the presynaptic, this doesn't prove this is presynaptic inhibition. But a variety of pharmacological studies that I won't go through support the idea that this is mediated by GABA and not by glycine, and has all of the pharmacological features of presynaptic inhibition. But in a sense, the most rigorous test of presynaptic inhibition is this idea of a reduction in transmitter release probability. And some time ago, my colleague at Columbia, Larry Abbott, developed this quantitative model that on repetitive stimulation, the suppression of EPSP or EPSC amplitude reflects presumably the depletion of vesicles from nerve terminals, and he defined a mathematical model that monitored instantaneous release probability, the vesicle pool size, and the number of vesicles released, and derived a quantitative model that applies to most central neurons there. And Andrew Fink has been working with Larry to take this model and see whether this model provides increasing evidence that the suppression of EPSC amplitude is mediated by a reduction in transmitter release. And so when you apply repetitive stimuli, in the absence of activation of the GABA preneurons, you see a decrement in EPSC amplitude. When you then activate these GABA preneurons, then the shape of this decrement decreases in way or changes in ways that's shown here. So these black dots represent the decrement with sensory stimulation alone, and with sensory stimulation where you're recruiting these GABA preneurons. And if you take these experimental data and fit that to the Abbott vesicle depletion model, what you find is that the theory very nicely predicts the decrement that you see following activation of these GABA preneurons. So this provides, I think, reasonably strong evidence that activation of these neurons is producing a suppression of EPSC amplitude through a reduction in the probability of transmitter release, in effect, a presynaptic inhibitory control. So then, at this point, GAD2 has provided genetic access to GABA preneurons, at least in the adult, and through physiology we can pick apart the actions of the GABA preneurons. And then we're reasonably convinced that activation of this set of inhibitory neurons mediates presynaptic inhibition, as defined classically by the studies of Eccles and others. So that, in principle, put us in a position to try and ask now if we can selectively manipulate these neurons, does changing presynaptic inhibition have any impact on motor behavior defined by these reaching tasks? So here we go back to the situation that we're trying to manipulate these GABA preneurons in the adult under conditions in which GAD2 Cre recombinase expression is selective to that set of neurons. And so just to remind you, we're doing these AAV injections at 8 to 12 weeks. This time we're injecting a construct that drives expression of GFP and a diphtheria toxin receptor. And the fluorescent protein expression nicely matches the distribution of GAD2 on sensory terminals. And so we can get about 75 to 80% expression of this viral construct in GABA preneurons with less than 1% expression in GABA postneurons. So we're now in a situation where in the adult we can target GABA preneurons with selectivity. So if we now inject or introduce the diphtheria toxin receptor, and then 2 weeks after introducing that, we introduce diphtheria toxin. In mice, diphtheria toxin is without effect because mice don't express the receptor. But when you genetically introduce the receptor, then introducing diphtheria toxin kills neurons within a couple of days. And so we can see evidence for that killing of GABA preneurons, because if we look now at sensory terminals before addition of diphtheria toxin, they're nicely studied with these small pink GABA pre-boutons. After administration of diphtheria toxin, you can see the sensory terminal that is now stripped away of its GABA pre-boutons. And so there's virtually 100% labeling post, there's less than 5% labeling here. And this effect is selective without impacting the GABA post neurons, because if we now do the same experiment and look at GAD1, GAD1 is also expressed in these GABA pre-boutons you can see here, but as well as in GABA post. After diphtheria toxin administration, the GABA pre-boutons are gone. There's no change in the number of GABA post neurons. So this gives us the ability to manipulate selectively this one set of inhibitory interneurons that are forming presynaptic regulation of sensory transmitter release and beginning to examine the impact on motor behavior. And so here, we revert now to this assay that Ayman Azim developed that I've shown you before, and this time we're going to add now some detail in terms of the kinematic analysis in pre-diphtheria toxin administered mice. And if we do that, we can, with this video analysis, monitor individual trajectories and then pool those trajectories in successive reaches. And what you can derive is a plot of the forelimb paw trajectory towards its intended targets under conditions in which the target pellet was seized or was missed. And what the important point here is that these trajectories are very similar, independent of hit or miss. So that means the initial stages of reaches are the same, and what's determining the success of pellet retrieval is some late event involving pronation or grasp. But the early phases of reach are relatively normal. So now what happens if we compare in the same mouse before and after diphtheria toxin administration these reaches? And so here is an example. So the first thing we've done is look at the consequences on diphtheria toxin administration on overall pellet retrieval success. And you can see that before diphtheria toxin administration, the animal is successful in retrieving a pellet about half the time. Soon after diphtheria toxin administration, a couple of days later, the success of reaching rapidly declines to 0. So there are no successful reaches under conditions in which GABA pre-neurons have been eliminated. And this raises the question, is this a wholesale, severe, general loss in motor coordination, or is this something that is somehow specialized to this reaching system? And one line of evidence that suggests there's some selectivity for this supposedly refined motor skill comes from looking at the animal's ability to perform other motor tasks that involve some accuracy of forelimb placement, walking on a horizontal ladder, for example. So these same mice that show this deficiency in reach do pretty well in terms of placing their fore and hind limbs, forelimbs in particular, on this ladder. And so the frequency of ladder rung misplacements is not statistically higher after diphtheria toxin than before diphtheria toxin. So this makes one think that somehow this GABA pre-system in terms of forelimb motor control is selectively affecting or perturbing this reaching system. So now here is this experiment that I introduced to you, the idea that in this same animal, before administration of diphtheria toxin, you can see that it's reaching with a relatively normal trajectory, even though a slight late placement error knocks the food pellet off this perch. But you can see here this normal trajectory. So now if we take the same animal a couple of weeks later after we've eliminated the GABA pre-neurons, how is its motor performance? And what I hope you can see is that there's a severe abnormality that this animal is moving, it's attempting to move, so the intent to reach the target is there, but in reality, every forward movement is associated with a reverse movement. It has enormous difficulties in actually providing a smooth reach trajectory towards its intended pellet target. And if we quantify this, you can see compared to this pre-toxin trajectory, there are these undulations and excursions and deviations in trajectory that I think you'll be able to see in the video analysis here. So Andrew, together with Ayman and Catherine Croce, have been analyzing this perturbation in motor behavior to try and understand its basic origins here. So if we plot here, this is just another way of showing the same data, velocity of forelimb reach versus position as a function of distance from the target. So this is early in the reach, this is late in the reach, This is pre-toxin, this is post-toxin. You can see a relatively consistent trajectory all the way through to the reaching and arrival of the target, whereas post-toxin, in the absence of GABA-pre neurons, you see these dramatic reverberations which reflect fall in limb reversals. So it's moving here forwards and backwards in a way that is quite abnormal and different, and you can quantify this here. Another way of demonstrating this is to plot velocity versus time. And what you can see here is that these deviations and excursions really represent oscillatory motor behavior here. And if we plot this from mouse to mouse, what you find is that there's consistency in the motor oscillation that is induced by eliminating the GABA-pre neurons. And that oscillation has a peak frequency of around 20 hertz in many, many, many mice here. So somehow taking away the GABA-pre system has induced these motor oscillations that have this dramatic behavioral effect in terms of forelimb movement. And so there is one major conclusion and then one set of analyses that I want to share with you. So the major conclusion here is that presynaptic inhibition is essential for this aspect of motor control, despite the persistence of all of these postsynaptic inhibitory circuits. If you take away presynaptic inhibition of sensory input to motor neurons, the entire spinal circuitry involved in motor control is unable to cope with the loss of presynaptic inhibition, the loss of that gain control, and you see the consequences in terms of motor behavioral defects. So in a sense, this provides a validation to the studies of Frank and Fortes and Eccles, arguing that presynaptic inhibition is a specialized anatomical physiological function, and now we see for the first time its behavioral counterpart. But the second thing we wanted to understand is why, when you take away presynaptic inhibition, do you get these marked reverberatory oscillations? And Andrew has been working with Larry Abbott to try and think about a model which could account for the oscillatory motor behaviors upon removal of presynaptic inhibition that is related, in a sense, to the idea that presynaptic inhibition is a form of gain control at these sensory motor synapses. If you take away presynaptic inhibition, then the sensory gain increases. And this forms part of a model which considers the impact of loss of presynaptic inhibition on the functional properties of two sets of motor neurons and their recruited muscle, which serve antagonistic functions at a particular joint. So I want to walk through that model a little bit. So this is an analysis of joint angles here at a particular limb joint here, where we're applying, in this case, an extensor torque. So what will happen is the activation of these extensor motor neurons will activate extensor muscle and will increase the theta, the angle at which this joint is placed here. And if we do that, and we can quantify this in ways that Larry has done, if we do that, then as we increase theta, as we extend at this joint, there will inevitably be a consequent compensatory activation of the stretch receptors of the antagonist flexor muscles. That's what this indicates here. So this antagonism there presumably activates the proprioceptive sensory feedback system that is coming in and providing excitatory input to the antagonist flexor motor neurons. And so in this view, one function of presynaptic inhibition in this system may then be to exert an inhibitory control on the compensatory sensory input that would be activating the antagonist flexor motor neurons. And this thing sort of got a little ahead of me here. So I want to show you the indication of what happens in the presence of presynaptic inhibition, this smooth extension of that joint, because presynaptic inhibition has effectively suppressed this compensatory sensory input to antagonist motor neurons. But now what happens in this model if we take away presynaptic inhibition, increasing sensory gain? Then in contrast to this, what you find under conditions where sensory presynaptic inhibition is blocked is the system begins to reverberate, because now this system activates the flexor, will then activate the compensatory extensor sensory feedback, and the whole system oscillates between this reverberation driven peripherally by the activation of compensatory stretch receptors. So this model, in a sense, does a reasonable job of explaining before and after removal of presynaptic inhibition, the angular velocity, which looks quite a lot like the experimentally derived data before and after removal of GABA preneurons, where you get this oscillating behavior. So the other question that we've been interested in is why in these mice, when you take away presynaptic inhibition, do you see this 20 hertz oscillation? What is the significance of oscillation at that particular frequency? And one way of thinking about this that derives from this model is shown here, that if you actually plot sensory motor gain as a function of frequency derived from the model, the slope of this line is relatively shallow. So under conditions of removal of presynaptic inhibition, where the sensory motor gain increases, a relatively broad set of increases in gain from 1.3 to 2.2 here, still, because of the shallow slope of this line, lead to a frequency change that is relatively narrowly windowed around this 20 hertz. So it may be that this 20 hertz oscillation here that you see in mouse after mouse after mouse reflects the quantitative nature of the relationship between sensory gain and output frequency. So these models here are helping us think about the consequences in terms of motor behavior of eliminating this one simple set of inhibitory interneurons. Now, I want to stress here that this idea about the model of consequences of gain changes in the sensory motor muscle reflex system is not new. It's a refinement of a model that Dick Stein generated some 25 years ago. The idea here mostly focusing on muscle, but the basic idea of Dick's work is really exactly the same, that changes in sensory feedback gain induce an instability in this sensory motor muscle reflex arm. So when gain is low, you get a simple response, and when you gradually increase the gain, you drive to this oscillation. So one of the remarkable things, I think, about these experimental studies using GAD2 as an entry point is we've generated experimental data that fit rather nicely some of the views derived theoretically by Dick Stein and Cappaday and others some 25 years before. So that supports the idea that what we've done here is selectively remove one gain control system from the spinal sensory motor circuitry in a way that was rather faithfully predicted by the theory. So it supports the idea that other extraneous impact of removing these neurons is unlikely to be true. So this leaves many issues unresolved about the role of presynaptic inhibition. I think it does support the idea that there's an essential function for presynaptic inhibition in motor control in terms of these skilled reach behaviors, but leaves many other things unresolved. And I just want to discuss some of the implications of this work in the context of what we're trying to do next and other related thoughts in the field. And so this is the original image depicted by Eccles, where he identified these presynaptic inhibitory interneurons simply on the basis of physiology in a remarkably prescient manner some 50 years ago. And so the four things I want to talk about are the nature of recruitment and diversity of this presynaptic inhibitory system, since it seems to be important, what presynaptic inhibition loosely defined really does for motor systems, then why, since GAD2 has been so useful, why is GAD2 expressed with this selectivity in these presynaptic neurons? Is there any logic for that? And then I've described experimental systems. Could there be any clinical relevance of this presynaptic inhibitory system to things in human condition? So the big challenge that we have at the moment is that physiological studies have argued that you can recruit these presynaptic inhibitory neurons through sensory input, indirectly in the way that I showed, through local interneuronal input involved in central pattern generator circuits, as well as from the work of Eberhard Fetz in descending systems. So this, to us, raises the question of whether, even though I've defined this as a single GABA pre-system as distinct from its GABA post inhibitory interneuron counterparts, whether there's really diversity within the GABA pre-system that we haven't yet appreciated. And could there be perhaps 7 different classes of GABA pre-neurons, each dedicated to form presynaptic inhibitory contacts with different classes of sensory afferents, cutaneous and proprioceptive? A flip side of that question is whether these different systems, the sensory, interneuronal, and descending, engage different presynaptic inhibitory interneurons to build multiple microcircuits that use presynaptic inhibition. And Pablo Ruderman's work over the years has argued for a high degree of fidelity and specificity in the recruitment of presynaptic inhibition as a function of different peripheral sensory stimuli. So there's every reason to believe that there is going to be further diversity in this presynaptic inhibitory control system. And so to try and approach that, we've taken this anatomical viral approach. These days, because we can target these neurons with GAD2, we can express a recombinant form of rabies virus that jumps retrogradely into the inputs of these GABA pre-neurons, and in the middle of now trying to define, by virtue of GFP expression, the diversity of different classes of neurons that converge on these GAD2 defined neurons. And if we can begin to understand that, then maybe we can manipulate selective inputs into this GAD2 GABA presynaptic system and look at the individuality of recruitment of this set of neurons. The second thing I want to emphasize is that throughout this talk, and in a sense behaviorally, we've equated the GABA pre-neurons with this phenomenon of presynaptic inhibition. But it may be more accurate to refer to this system as a gain control system, if you like, a sort of rheostat for sensory motor transmission. And there's several lines of evidence that lead one to think that under certain circumstances, activation of this set of inhibitory neurons, because of this strange ability of these neurons to depolarize the sensory terminal, because of the strange chloride equilibrium potential, because KCC2 is not expressed in sensory neurons, that you can get multiple effects according to physiological context. And I want to give you 2 examples of that. The first comes from experiments that we did, that echo experiments that Eccles did many years earlier in the cat, that show that under some conditions, recruitment of this GABA pre-neuronal system, actually the depolarization of the sensory terminal, is sufficient actually to release glutamate and activate the motor neuron. So this would be a presynaptic facilitation or excitation. And one of these variables is the temperature in which the preparation is maintained. And so if you perform these optogenetic activation experiments at 24 degrees and record from the motor neuron, you see a massive EPSP recorded from the motor neuron at low temperatures, whereas at high temperatures, you perform the same optogenetic activation, and you see only an IPSP because presumably you're recruiting the GABA post. And this plots the EPSP amplitude recorded in motor neuron as a function of temperature. So we don't know the significance of this temperature-dependent activation, depolarization, release of transmitter, but it indicates that this isn't an obligate inhibitory system, and some systems can activate these neurons. And the second comes from the sort of modeling experiments, where as you apply repeated pulses and control the probability of transmitter release, there are conditions in which you can apply GABA agonists, presumably mimicking the effect of recruitment of these presynaptic inhibitory neurons. And instead of seeing synaptic depression in the presence of muscimol, you see synaptic potentiation with repeated sensory stimuli. So these 2 sets of experiments together sort of begin to raise the possibility that this set of neurons is really exerting a much more sophisticated gain control on sensory motor transmission, that the incidence of sensory stimulation combined with the number of prior trials is all feeding into the impact of these GABA pre-systems on sensory motor transmission. We're trying to look at that in more detail. So most of the experiments that we've done have been based on this genetic entry point through GAD2 expression. So GAD2 is the second of these GABA synthetic enzymes, along with GAD1. And there's good evidence that GAD2 expression increases the level of GABA that an individual neuron synthesizes. So one of the functions of GAD2 might be ensure that these presynaptic neurons contain high levels of GABA, and that might be necessary because of the strange synaptic arrangement. You don't have an entire postsynaptic neuron to form contact with. You've got that single sensory terminal that you're trying to inhibit transmitter release. So levels of GABA may be important, but there's also this idea that, or the possibility that GAD2 does other things in terms of the functional relevance of presynaptic inhibition. And again, if we go back to Eccles' studies in the 1960s, one of the things that Eccles found by recording from these presumed GABA pre-neurons is that they could sustain enormously high firing frequencies following sensory activation. Here, they could sustain 100 hertz or more firing frequency. So it may be that under conditions that you recruit the GABA pre-neurons, you have to release a lot of GABA under these high-frequency firing conditions. And this becomes relevant from studies that Tien and Nickel performed some 10 years ago, looking not in the spinal cord but in the hippocampus at the impact of GAD2 on sustained inhibitory transmitter release at high stimulation frequencies, because they showed that in GAD2 null mice, the ability of these neurons to sustain GABA release at high frequencies was dramatically abrogated. So GAD2 is binding to the synaptic vesicles and somehow is involved in presynaptic vesicle mobilization. So in that view, it may be that GAD2 is necessary in these presynaptic inhibitory neurons, not only to boost the levels of GABA expressed in the neurons, but also to permit those neurons to sustain GABA release under high firing frequencies that are necessary to achieve presynaptic inhibition. So this then raises the issue, does GAD2, does targeting GAD2 have any relevance beyond these experimental systems? Could it be relevant to motor performance in humans? And in this context, there is a neurological condition called stiff person syndrome that begins to suggest that the GAD2 system in spinal cord that I've described may have relevance to motor coordination in humans. So in stiff person syndrome, individuals have their limbs extended, they lose the ability to flex and extend in a controlled manner, and they're severely disabled because of this co-contraction of extensor and flexor muscles under conditions of limb extension. It turns out that some 70 to 80% of individuals with stiff person syndrome show autoantibodies against GAD2. So the idea is that somehow GAD2 antibodies are getting into this system and preventing presynaptic inhibition in spinal cord and elsewhere. And under these conditions, you see in forelimb movement this co-contraction of biceps and triceps. So it may be that in part what we've been seeing in terms of the GAD2 manipulation of this presynaptic inhibitory system has some relevance in clinical neurology. So with this example, I just want to sum up and point out that as you take this systems-level view of motor systems, what we've been trying to do is to pick apart the logic of circuitry in spinal cord, essentially one interneuron at a time. And I gave you a very brief overview of some of Iman's work on proprioceptive systems. I talked about this presynaptic gate. We're looking at some of these postsynaptic inhibitory neurons and finding impacts on motor behavior through each of these manipulations. And the long-term goal is to use these genetic entry points together with the classical physiological understanding of motor systems to try and work out the underlying logic of motor control, linking behavior to neuronal system to individual neuronal identity. And I will stop there and just emphasize the work that Ayman Azim developed this reaching task and performed the work on propriospinal neurons together with Bror Alstermark's group. Andrew Fink did pretty much all of the work on this presynaptic inhibitory system, benefiting from mice generated by Josh Huang, ideas and theory from Larry Abbott, and experimental studies of behavior from Catherine Croce. And so I will stop there and just acknowledge the many people who funded the work in the lab, and thank you for your attention.
I'd like to express our most sincere thanks for you taking the time to come and present this elegant work to us. We all know that the spinal cord has provided a window to the rest of the brain for many, many years in helping us understand how output neuron control is established and how circuitries interact to direct that control. And it's obvious that with your new optogenetic and with your molecular tools here that you're going to continue to lead the way and show us how the brain really works. So thank you very much. And with that, I'd like to present you with this little memento from the Physiological Society, the IUPS TP Feng lecture for 2013. Thank you very much.
Thank you.
Gonna pay extra to get that home.